This is your third and final video for photosynthesis, and we're going to talk about the Calvin cycle. So remember from the light reactions in the thylakoid space and the thylakoid membrane, we made NADPH and we made ATP. So here's where the rest of the reactants come into play. Remember that you have the diffusion of CO2 coming through the stomata into the plant cell and into the stroma of the chloroplast. That's where we are right now. We're in the stroma of the chloroplast, which is basically the cytoplasm of the chloroplast. And we're going to start with this molecule right here. It's called ribulose biphosphate. Ribulose biphosphate. And this is going to be a CO2 acceptor, and we'll see what this means in a second. This is a CO2 acceptor, and it is a 5-carbon molecule. So 1, 2, 3, 4, 5. Each one of these circles is a carbon. And in order for the math of this all to work out, we have to run this reaction in triplicate. We have to run it 3 times simultaneously. Just go with me on it. It'll make the math work out and it'll make it easier to accept. So because we're doing this 3 times, we're going to have 3 of everything. So we're going to have another ribulose, 1, 2, 3, 4, 5, biphosphate, and a third ribulose biphosphate. And again, this is a biphosphate. So really on the end of these, there's a phosphorus or a phosphate group. So 2 phosphates per RUBP. And I'm not going to draw them past this point. I'm really only going to include them here. All right, so there's your 3 RUBPs, your ribulose biphosphates, for a grand total of 5 times 3. 15 carbons. 15 carbons. There's an enzyme called Rubisco. So we're gonna go through the cycle. Rubisco is probably the most prevalent enzyme in the world. It's probably the most prevalent, if not the most prevalent enzyme, one of the most prevalent enzymes on Earth, just because of its role in this cycle. Rubisco is going to help us, or I guess help plant cells or anything doing this cycle, fix carbon. And what we're going to do, or what they're going to do, is add a carbon dioxide molecule that's diffusing in from outside the cell. We're going to attach it onto each of these ribulose biphosphates. So we're going to need 3 of them, because remember, we're doing this in triplicate. We're doing this in sets of 3. So we're going to take 3 carbons from carbon dioxide, and we're going to tack them on to the end of these ribulose biphosphates. Remember, this is a CO2 acceptor, so it's gladly going to accept these CO2s. So here's 1, 2, 3, 4, 5, and if you want, go ahead and change color. You can make this last circle red or something to show you that it's coming from a different source. It's CO2, and you can actually even make these CO2 molecules red if you want. 1, 2, 3. Remember, each of these ribulose biphosphates gets an extra carbon from carbon dioxide. So now we have 18 carbons. We have 3 6-carbon molecules. All right, remember that these are packed with phosphate groups, and phosphate groups have a negative charge, so these are actually pretty unstable. And what's going to happen is these are actually going to end up splitting. These are actually going to end up getting split. And we're going to end up with 6 3-carbon molecules. One, 2, 3, 4, 5, 6. So we get 3 on this side, 3 on this side. And remember, if you're color coding the CO2s, maybe like this one is red, it came from a CO2, maybe this one is red, it came from a CO2, and maybe this one is red, it came from a CO2. So we're not getting rid of any carbons here. We still have a grand total of 18 carbons, but because we split each of the ribulose biphosphates, they're now rearranged into 3 carbon atoms, and you get 6 of them. All right, at this point, these 3 carbon molecules are called PGA. That's kind of like an extra little tidbit. You're never really going to have to use it, but if you ever see it in your textbook, that's what these molecules are. And at this point, and I'm really just going to go with the bare minimum here, 6 ATPs are going to come in, drop off a phosphate group, and leave as 6 ADPs. So 1 ATP, ADP, 1 ATP per molecule here. All right, so there's 6 total. And 6 NADHs, sorry, 6 NADPHs are going to come in and drop off an electron, which is going to help it form a hydrogen, take up a hydrogen, and they're going to leave as 6 NADP plus. Remember, 1 NADPH per molecule, so that's why we have 6. Right? This is called the reduction phase. We're reducing each of these 3 carbon molecules by adding a hydrogen. This is why we need the electron carrier. And we're just rearranging some of the bonds, and that's going to take some energy. So this is why we need ATP. What that's going to do is generate 6 G3P. We've actually made G3P at this point. Remember, G3P is just a 3 carbon molecule, and it's the same G3P as in glycolysis, as in cell respiration. So this is a very familiar molecule. What are the odds? And I'll tell you, they're evolutionarily related. It's not a coincidence. So we've made 6 G3Ps. So I'm going to go ahead and redraw those. And at this point, one of the G3P molecules is going to leave the carbon, or sorry, leave the carbon cycle, leave the Calvin cycle. One G3P is going to leave the Calvin cycle. The remaining 5 continue on because they need to replace ribulose biphosphate. So at this point, now we're kind of in step 3. We just have to take these remaining 15 carbons, rearrange them so that we get our 15 carbons that we started with, basically. So there are a series of enzymatic reactions that do this. Remember, the whole point is to regenerate Rubisco, to regenerate RUBP. That's just the abbreviation for it. And to do this rearrangement, we need 3 ATP to come in, drop off their phosphate, and leave as 3 ADP. And then that will restore our baseline, and we can start again. We can do this whole thing over again. So it's actually pretty straightforward as far as cycles go. So at this point, let's do a quick tally. Let's see how many ATPs and NADPHs we've made. So let's go ahead and count, or how many we've used to make this. So 1 G3P, 1 crank of this triplicate cycle, has cost us 6 NADPHs and let's see, over here we had 6 ATPs and we had 3 ATPs, so 9 ATPs. this entire thing happens again. Oops, this entire thing happens again. Because basically where we're going with this is we're going to take two G3Ps, and with an enzyme, we're going to link them up and make glucose. So all of this has to happen again. So I'm just going to say times 2. And that's going to get you 12 NADPHs and 18 ATPs. This is the same 12 and 18 that we talked about in the last video. That's going to get you another G3P, and then your 2 G3Ps, your 3 carbon molecules, using an enzymatic reaction, can combine to make your 6 carbon glucose. And at this point, let's briefly talk about what cells do with glucose, because plants have mitochondria too, and they need to burn glucose for ATP themselves. So at this point, the glucose can be used as a raw material to do a variety of things. It can be used in cell respiration in the mitochondria. It can be used to construct cell wall, cellulose, right? It can be used to build all of the other structures the plant needs. It needs to build lipids, it needs to build proteins, and it needs to build nucleic acids. And the base for this, the raw substrate for this, is these glucose molecules, these carbon-rich, hydrogen-rich molecules. If you're making proteins, just remember that proteins have nitrogen and sometimes sulfur in them. So the nitrogen and sulfur have to come from the roots, in addition to the glucose being made here. If you're making nucleic acids, not only do you have nitrogen, but you have phosphorus, right? You can't make everything with glucose because it only has CHO. So you do have to use material from the roots coming in. But let's go over this cycle one more time, and then we'll end the video. So you've got NADPH, and you've got ATP coming in from the light reactions in the thylakoid space, in the thylakoid membrane. All right, starting with this molecule of RUBP, ribulose biphosphate, Rubisco, which is an enzyme, is going to attach a carbon in the form of carbon dioxide to the end of each of these. So we're going to run this in triplicate. That's going to get us 3 6-carbon molecules. They're going to get split using ATP, and they're going to get reduced using NADPH into 6 3-carbon molecules, one of which exits the Calvin cycle entirely as G3P. The remaining 5 G3P are going to get rearranged using some ATP, and we replenish our stock of ribulose biphosphate, and this cycle can go over and over and over and over again. So if we zoom all the way out, we've made glucose. Let's keep track of our substrates and our products. So we had light as a substrate coming in and exciting electrons. We had water coming in as a substrate, which donates electrons to replace the ones lost in photosystem 2. We had the reduction of electron carriers and the synthesis of ATP that was used to rearrange carbons getting linked together with Rubisco, and we get those carbons from carbon dioxide. And some of those molecules that we make exit the Calvin cycle as G3P and link up to make glucose, right? So technically, the product of photosynthesis isn't glucose. Technically, the product is G3P. And then afterwards, you just put 2 G3Ps together to make glucose. But that's the big picture. I hope this helped, and I'll see you guys in class.